Hello and welcome to another episode of the Omics cast. My name is Albert Vilella, and today we're going to talk about the Human Cell Type Methylation Atlas. As usual, for those of you that want to follow me on Twitter, my Twitter handle is can be followed at twitter.com/Albert Vilella, all one word. I'm happy to answer any questions for you on LinkedIn. Here are my LinkedIn details, and this slide deck is available upon request. So today I wanted to talk about a paper that was published late last year, and it's a very comprehensive resource data set that they've generated in generating a methylation atlas for collection of cell types and tissues, including data that gives us information about circulating cell-free DNA. So they did a methylation, they performed a methylation atlas of 25 human tissues and cell types. And from these, one of the main results was that the plasma cfDNA of healthy donors originates from white blood cells, erythrocyte progenitors, vascular endothelial cells, and 1% of hepatocytes. So the cfDNA that we have in our bloodstream, outside of the cells, is 99% or 95%. In this case, related to blood cells, white blood cells, erythrocyte progenitors, or the lining of our vessels, vascular endothelial cells. They also, in this paper, they did analysis on non-healthy individuals, including sepsis, islet transplantation, cancer of the colon, lung, breast, prostate cancer, and others. How did they generate this data? Well, they used a technology that's been available for about a couple of decades, 2 or 3 decades, which is microarrays. And in this paper, they used Illumina microarrays that, if you zoom in, they look like this. If you zoom out, they look like this chip here. And these microarrays, most of their market share, most of their usage is for genotyping, to find out the genotype specific positions of the human genome, as it's shown here. So they basically by two fluorophores, one green, one red, can determine if at a given position you're a home for green, a home for red, or a heterogeneous green-red here. So for this SNP here, it would be CC, and for this SNP here, it would be GA, and for this SNP here, it would be TT. And these fluorophores, green or red, is what is detected by the machine that reads these microarrays. In the world of epigenomics or methylation, what you are measuring is the presence of a C or a T in CpG position. CpG means a C followed by a G in the human genome, which is a dinucleotide that can be marked epigenetically by a methylated C or other marks. And what they did in this analysis is to look at the methylated or non-methylated Cs in these CpG positions. And they do it by, similarly to the microarrays for genotyping, measuring after bisulfite conversion, which converts this methylated C, keeps the methylated C as such, but converts this unmethylated C into a UG, which after amplification is turned into a TG. They hybridize it into array, and then they measure these two alleles, methylated or unmethylated. And in this paper, they used the newer of the arrays for methylation that Illumina has, which is called the EPIC array, which has about 850,000 of these positions in the human genome that have been carefully curated to be meaningful. The previous array, the 450K array, had about 450,000 positions, and it's been discontinued in the last year or two by Illumina. So Illumina now wants everybody to use the EPIC array and only has a few of the remaining 450K arrays for people to complete their studies. So where is methylation in the human genome and how does it change? This is a schematic also in part of the Illumina website that shows here a gene body conformed by exons and introns, etc., and how in the promoter area upstream to the gene, there can be CPG islands or accumulations of CPGs. That can be hypermethylated repressing transcription. Next to this island of concentrated CPGs, there's what's called the shore. Next to the shore is the shelf. And there can be also hypomethylation of these regions. And then outside of these genes and promoter regions, then there's the heterochromatic part of the genome that can also be hypermethylated. If it's hypomethylated, it leads to genomic instability, etc., etc. So basically, by measuring the methylation marks at specific places in the human genome, we can tell about the functional state, the cell type specificity of these nuclei, these cells. And 5-methylcytosine is one of the many different modifications on the nucleotides that can take place. And this is a very long list here. So this is in the C portion of the ATCG. As you can see, 5MC is here, 5MC in white is here, next to 5HMC, 5FC, 5CAC, etc., etc. And there's lots of them, many possibilities. This graph of information contained inside is part of the DNA Mod website on the Hoffman lab. People in Canada, Michael Hoffman and his lab that work on this. And 5-methylcytosine, chemically, the structure is like this. And then there's many ways of measuring it. And there's a very long list of these in the paper, in the website, in the DNA Mod website, the Hoffman lab, citing the papers of the different methods that people have tried, including OXBS by Michael Booth, who was one of the people that started the work that ended up being the first proof of concept with which Cambridge Epigenetics, the company, started doing the work. And then the last one probably in this list, unless I'm mistaken, is TAPS, which is a method published this year by Liu and others. There's another method that was published this week, which is from NEB, New England Biolabs, which is called EM-seq. And EM-seq is a alternative to the conversion by a sodium bisulfite here, which starts in a molecule like this that has methylated Cs or hydroxymethylated Cs. It converts everything that's not methylated or hydroxymethylated, and then it gets sequenced as this. This leads to quite a lot of loss of material, so you have to start with quite a lot of DNA if you want to do this. And the EM-seq method, together with the TAPS and TAPS beta method we saw in the previous slide, are alternatives that are enzymatic alternatives to these that use, in case of the EM-seq method, TET2 plus an oxidation enhancer, followed by another enzyme, ApoBec. So it's an enzymatic way of converting the material so that when it's sequenced on an Illumina sequencer, you can find out about it. Obviously, if you use other methods that can give you a signal direct from the molecule without any conversion, like the Oxford Nanopoint method, then you don't need to convert it into anything to be able to find out the methylated Cs in CPG context. But as I was saying, in this paper, what they did is the old school way of using the epic array for this. And this requires quite a lot of materials we will see later on in the details. So this is a graph that basically shows the sorting, the clustering of tissues by their methylated status. And basically, they've chosen close to 8,000 CPGs, 7,890 CPGs that stretch out, that differentiate the different tissues of all the 850,000 that were assayed. And they are either methylated or unmethylated here, unmethylated in blue, methylated in yellow. And basically from the material, they performed the array, and then they did this clustering, this separation into tissues. And as you can see on the left-hand side, most of these are things that look like blood, right? So like monocytes, B cells, T cells, natural killer cells, more T cells of CD8 plus T cells, neutrophils, erythrocyte progenitors, right? And then the rest here are an assortment of quite a wide variety of other tissues that they profile here. And they generated this atlas basically of methylation types, of methylation marks across all these tissue types. So what do they see from this? Well, what they see in middle is that from healthy individuals, if you look at the cfDNA, most of the cfDNA, most of the circulating DNA that we have in our plasma, that's circulating free in our blood, not inside cells. Most of it is composed by things that look like blood, right? As it's shown here is this pie chart, and in a collection of bar charts here from males and females of different ages. And so the biggest contributor here is blood cells itself. That's what contributes the most to the circulating free DNA that we have in our serum, in our blood. What can happen in cases of disease or different conditions, like here the transplant of this diseased individual, I think there's more than one, 3 recipients of this transplant. Well, in this case, this fraction of cfDNA gets altered. And as you can see here, there's a bunch of pancreatic DNA that comes from pancreatic cells that is now in the bloodstream. And that's not normal. That's not what happens in the healthy individuals. And as we saw in the previous slide, but you can see it very clearly. There's a lot of evidence of it in these people that have undergone a transplant. A different case, sepsis. What does alter the cfDNA content in the case of sepsis? Well, lots of hepatocytes, like hepatocyte marks here. And also they did this correlation with alanine aminotransferase expression, which is a standard biomarker for hepatocyte damage. So again, measuring the DNA in our bloodstream outside of cells in different conditions, they can measure different contributors to this DNA in these different conditions. And then cancer, very important to be able to detect cancer early. In case of colon cancer, can we see colon cfDNA, cfDNA contributed from colon cells in colon cancer? And the answer is yes. For colon cancer, we see lots of colon cfDNA. We also see a little bit of breast cancer DNA. For breast cancer, most of it is breast. For lung cancer, most of it is lung. Out of what we don't see, which is the background, essentially the blood cells. And for non-identified cancers of unknown primary, there's always one of these bars rising up, which could be an indication of what type of cancer this is. And so as you can see very clearly, just by taking a blood sample and then extracting the plasma DNA and then measuring it in an epigenomic manner like is shown here, you can get an indication of the diagnostic value of what type of cancer that individual may have. So as I say, in the methods, all this was done with methylation arrays. Either the 450K, they took some data from previous studies, or the EPIC array, which is slightly bigger, 850K, 850,000 CpGs. How much material do you need for each of those? Well, quite a lot. You need about 250 nanograms of DNA in about 10 mils of blood. Once you've spun it twice and then you extract the cfDNA from the plasma, you can obtain maybe 5, 6, maybe if you're lucky, 8 or 10 nanograms of DNA, and that's 5 mils of blood. Sorry, 10 mils of blood. That's the size of a lipstick. So to get about 25 times more than that, or 50 times more than that, you need to extract a lot of blood from the patient. What did they do with that? Well, for some of these tissues, not blood cells, but tissues, they dissociated them and captured them using anti-human CD105 magnetic bits from Miltenyi and some other ones from Miltenyi here to do the dissociation, basically, then extract the DNA outside of it. For the blood samples, they usually drew about 20 mils of blood, so that would be about the size of 2 lipsticks, if you want. And very quickly, within an hour or so, spun to separate the plasma, and then they stored -20. It's recommended to store -80 if you have to keep it on the move for several hours, but here they did -20. And then the samples that they processed through methylation array, because they only did about 20 mils per individual, they had to pool them, right? So what we see here is not a per individual assay. It's we see samples from pooled individuals, several individuals, out of which they only took about 20 mils, maybe 40. And so DNA was extracted from 8 mils of plasma, and samples were added until 250 nanograms were reached. And depending on the individual, some of them will have more or less cfDNA in their same volume of plasma, but they said between 7 and 19 samples were needed per pool. So you make the calculations, they need to extract quite a lot of DNA, quite a lot of blood per pool, right? DNA extraction, 250 nanograms collected, cfDNA extracted via the Kaya Symphony liquid handling robot. There's several in the market, Kaya Symphony being one of them. PerkinElmer, Hamilton, they all have their own automated ways and offerings to do the plasma cfDNA extraction. Data processing, because these arrays, methylation arrays, they need to be processed as such, and there's an R package called MinFi for that in R that will allow you to do that, and then they filtered for CpG's positions represented by no less than 3 bits per array, and then they filter the ones that had less than 3. And then the p-value of the luminescence detection threshold was 0.01. They performed the pre-process Illumina function to filter out this background. They compared the 2 arrays, the EPIC and the 450K array, and they used the MAE to discard the CpGs that weren't compatible between the 2. And for reproducibility, they also tested how low can they go, how low could they go in terms of amount of DNA needed to start the process and get sensible results out of it. And because they said they started with 250, they also tried it down to, in the titration, down to 50 nanograms. And 50 nanograms is still a lot of cfDNA if you need to extract blood from someone to update 50 nanograms of cfDNA. What they show in this titration from 50 to 100 to 250 is that the profiles change. In this case here in red, they see more hepatocyte marks at the lower end. If this is true or not, it's difficult to say, but it changes if you do it with 50 than if you do it with 100, 250, or the original 250 here, these two being replicates. It changes a little bit in the other one. So this titration, I don't think they can claim that it was perfect result, in the sense that the amount of DNA you start with does matter for these methylation arrays. Now, this is what what I wanted to highlight from this paper, basically the technology that they used, what they got out of it, and how we've learned in the last few years what the cfDNA in our blood is and what's important, what can we learn from it so that we can do things like cancer detection from it. As usual, you can follow me on Twitter, twitter.com/Vilella. I don't mind at all if you ask me questions on LinkedIn. You can contact me on LinkedIn and I'll answer any questions you may have. And this slide deck is available upon request. I hope to catch up with you in the next episode of the Omics Cast. Thank you for watching.